For those of you who are new to the Stedman approach, this will just be review, all old slides, stuff that I think I've done a crummy job of explaining already. Yes? Scantrons on Monday, yes. There will be one extra page that you will have to turn in that will be shortish answer stuff. The blue Scantron, yes, the big blue one. And remember to put your student numbers on it. I haven't taken points off for people putting them in incorrectly, but I'm thinking about it. So it's really annoying when you're trying to input grades and those numbers don't match. So make sure you have your student number with you. Second thing, much more important, at least as far as my undersized ego is concerned. How many of you listen to This Week in Virology? I know a few of you do. We got not one, but 2 shout-outs in the last issue, which was pretty cool. So I sent them an email, so maybe we'll hear something more. But today they're doing a special live version in DC. For what reason? Science March, or March for Science, tomorrow. No requirements whatsoever. You don't get any extra points if you show up. But we'll be over in SRTC starting about 9:15. Heading over to the March site down on the waterfront. So I think it should be a lot of fun. We shall see. Hopefully, cross fingers, et cetera, it's not going to pour with rain. So we shall see. Even if it does, it's Portland, so we don't melt, right? Okay, so that being said, let's move on with what we've talked about for the last 3 weeks already. How did that happen? So a couple of things to really bear in mind and stuff when I was going back and looking at this, I know that I hadn't done a terribly good job of explaining. So yeah, the virion is just the extracellular particle, but there are a couple of things about that that I wanted to talk about in more detail, particularly having to do with these enveloped viruses, which we're going to spend a lot more time talking about later, don't worry. Having to do with the envelope and the envelope glycoproteins. Of course, these are all cellular lipids. But very often inside these enveloped viruses, there's some kind of icosahedral and actually sometimes a helical capsid, or a lot of people will also call these are nucleocapsids because they have nucleic acids inside them as well. And so we'll talk a lot more about these kinds of virions later on. So you can definitely have a capsid even in an enveloped virus. And not surprisingly, for geometric reasons, these are often have icosahedral symmetry or some kind of helical symmetry. And by the way, stop me, ask questions as we move along here. Yeah, Ian, I knew I could get you.
Just waiting for you. So, will you ever have an enveloped virus that doesn't have a capsid?
Will you ever have an enveloped virus that doesn't have a capsid? This kind of gets to the definition of what a capsid is, or a nucleocapsid. Nucleic acids are always associated with some kind of protein when they're in the virion. Whether you would necessarily call that a capsid, I'm not entirely sure. When we talk about flu, we'll talk about the individual segments, each of which is packaged with protein. Is that segment a nucleocapsid or not? Because there are 8 of them. So it starts to become a little questionable. But the main thing is that the nucleotides are always associated with protein in some way. Okay, so what is a virus? Lots of different definitions, but again, the one that I like is ridiculously ancient. You know, I was born before then, but genomes, nucleic acid, yes, replicating inside living cells with cellular synthetic machinery. And one of the things I think I didn't do probably a really good job of pointing out, and it does bear repeating, is that the main cellular machinery that all viruses need is the ribosome. All of them need ribosomes. All of them need cellular ribosomes in order to make proteins. When we find a virion that's got an encoded ribosome in it, that may change. But at least for the time being, that's really the point. And that also kind of gets us to the whole idea of the Baltimore classification, where you're looking at the messenger RNA. So not surprising, the messenger RNA is really important because that's that real cellular machinery which is absolutely required for all of these different viruses. And then, the specialized element that can transfer the viral genome, what's that? Also name is the Virion, exactly. So these virions on the outside. So this, I think, is a nice inclusive definition of viruses. And again, this is my homage to René Magritte, is that it's not this virion, which is the virus, it's this whole replication cycle, which is a virus. And it's not just the lytic cycle, I'm just using this as an example here. So the whole cycle of virion attaching, genome getting inside the cell, genome being replicated, genome being translated through messenger RNA intermediates or just straight RNA virus into more virions, and then going through this whole cycle. So this is really the life cycle, if we'll call it that, of these different viruses. Certainly, one of the defining characteristics of viruses is this extracellular element, the virion, which has an extremely unique shape relative to all other biological materials. There's nothing in biology that really looks like a virion other than a virion. And classic examples we've already talked about, head and tail, lots of icosahedral versions. This is a nice example of one of the plant viruses. Here's poliovirus that we'll talk about after the midterm. Here's that very first virus ever to be discovered as a small infectious particle, and then the pox viruses that we'll talk about a little later on. Lots of diversity, but most of it around this helical or icosahedrally symmetric shape for purely geometric reasons. Viruses follow the laws of math just as all the rest of us have to. So counting viruses, the key here is really the Plaque assay. The Plaque assay is by no means the only way that you can measure the amount of virus that you have in a particular situation. Animals are a great way of looking at literally the whole infection process. If it's just a virion and it doesn't actually infect that particular organism, who knows? So animals definitely tell you something about infectivity. Of course, whole animals also have defense mechanisms, so exactly how much you have there. But if you really care about viral disease, just looking at cells, unless it's a cellular virus like a microbial virus, is really not going to tell you very much. So if you're really interested in viral disease, you really have to look at a whole animal system. Electron microscopy is amazing. You get to see these beautiful pictures of these things, but it's just looking at virions. It tells you nothing about infectivity whatsoever. So, and as we've talked about a little bit, and we'll talk about much more as we move on, there are plenty of viruses that make non-active virions that under the electron microscope look exactly like they should be infecting like mad. So great, wonderful, these amazing structures that we can get from electron microscopy or some of the other techniques. Electron microscopy usually works much better than most of the rest of them. If you want to look at infectivity, yes, you can use animals. Animals are really, really expensive, and they've got all these extra defense mechanisms. So the classic method, and this is what we use for all of the bacterial and archaeal viruses, are the Plaque assay. And so this is the whole idea of the Plaque assay. I was literally getting ready to do one of these right before I came over to class today, which is partly why I've got all my gear with me. So idea here is you dilute your virus. And the key here is that usually the virus, and in this case it's going to be your virions, are usually present at very, very, very high concentrations. So if you were to just try and do a Plaque assay with this undiluted stuff, what would you see, Alice? Clear, or TMTC, too many to count. So you have to do these serial dilutions. And then from each of these serial dilutions, you make a number of plates which hopefully will have a countable, not a too many to count, number of plaques on them. And each of these plaques represents a original infectious virus that's now replicated many, many times and infected all of the cells around that particular cell. And so you can count the number of plaques on your duplicate plates, or in this case, quadruplicate plates. Take the average of the number of plaques here that tells you the number of viruses that you had in this tube. And then you can back calculate from your dilution to how much you had here at the very beginning. This is really, really, really easy math. So you shouldn't need a calculator for this. In fact, you're not allowed to use calculators for the exam. But I might ask you something like this. Hopefully, this is something you can do by hand. Yes? No? Good. Okay. Any questions about this particular aspect of things? Plaque assays are really critical. And again, we do it all the time. Yeah?
So for some of the assays to count viruses,
Mm-hmm.
we talked earlier about hemagglutination as being a semi-quantitative assay. Would you consider that as a method to count viruses or not?
So you can use hemagglutination. So the question is, can you use hemagglutination to count viruses as well? You can use hemagglutination to count virions as long as those virions will cross-link red blood cells. It tells you nothing about infectivity. But you can certainly quantify number of virions as long as they will agglutinate cells. So it is a way to count virions. It's not a way to count infectious virus. Okay, so this is just another quick example. These, as I think I mentioned before, are FIX-174, our friend the single-stranded DNA virus with this amazing conformational change making the tube through the membrane. Mix your dilution of virus here, bacteriophage, with your host cells, vast excess of host cells because they're the ones that are going to be growing here in the background. They've got to be something for the virus to infect in its multiple rounds of infection. And the agar to make sure that it doesn't get too far across this plate. It stops these things from all mixing with each other. And that's what I was doing. I was nuking the agarose this morning in the lab. So take this mixture, pour it onto your plate, then you let it sit. And in the case of the plaque assays that I'm doing, it's for about a week before you can then look and see where you have all of these plaques. Yes?
And then you would just multiply the number of plaques that you saw by the dilution, essentially, that you're at?
So I would multiply by the dilution, and if I want to get per milliliter, very often you use some fraction of a milliliter in each of these cases. In this example, it's in fact a milliliter which you put on each of the plates. Usually it's 100 microliters, so you'll have an extra factor of 10 here. Okay, and again, this is how I got that ridiculously large number of virions that you couldn't see when we looked at that tube beforehand. Another really important concept is this idea of the whole one-step growth curve. Because we can't study the replication cycle of one virus, one virion going through its one infection cycle, we try and look at a whole bunch of viruses undergoing their replication cycle in a whole bunch of cells at the same time. And the main reason there is that gives us enough to be able to actually detect. It's really hard to detect one virion. It's really easy to detect 10 to the 8th virions. So here you add virus to your host cells, but now at a vast excess of virus to host. So unlike what you saw with the plaque assays, now it's a vast excess of virus to host because you want to have complete infection. And that'll get us to the Poisson distribution in just a second. Yeah?
So it says add virus to concentrated host cells. Does that mean like concentrated virus to host cells? Because we're talking about a larger...
So the idea here is that you have this next step. And so if you're going to do dilutions, so one of the main things is once you've done that infection, then you want to make sure there are no free viruses around because they're going to mess up the rest of your experiment. Free virions, I should say. You don't have any more of the free virions around. So you're concentrated. If you do that dilution, then those virions have no chance to find more of the cells. So that's one reason to do the concentration step. And also, when you have a smaller volume, as anyone who's done plaque assays knows, it's way easier to deal with this than with the larger volumes. So, but it's also certainly concentrated virus. And the key, again, is to make sure you've got a vast excess of virus over your host so that you're going to have a synchronous infection. Okay, then, after you've done that, you mix them together, you dilute or wash so you don't have any further infections, and then follow the amount of virus which is produced over time. And so here, relative amounts of virus, and the best way to do this is with plaque-forming units, but if you're just looking at virions, you could use hemagglutination. You could even use electron microscopy to look at number of virions. Any way of actually detecting the number of virions produced on the y-axis. The x-axis is just time. And that's going to vary from virus to virus, how long it takes between the infection, which is what happens here, and production of infectious virions right here. Everything we're talking about in terms of molecular virology happens in this part right here, the latent period before any of these infectious virions are being produced. So all of the fun stuff molecularly that's happening as far as all of this virus replication is happening in this latent period. And I've had a number of people ask me about latent versus eclipse. Was that your question?
No.
Okay. What is your question?
If you're doing plaque assays over time, just to make sure that I understand this, you're basically going to have to set a set interval where you're setting up a plaque assay to watch over the course of, for example, a week. So every hour you set up a new plaque assay. Is that how you do it?
Yeah. So this is exactly right. So the plaque assays, because multiple rounds of infection, are going to take way longer than your actual one-step growth curve, because it has to be multiple rounds of infection. And so your actual experiment may be, in the case of, say, Phi-X174, half an hour. Because you collect a sample, start out the plaque assay with that, you put those plates in the incubator, and you'll come back the next day and look at them. And same thing is true with your 5-minute sample. You'll do a plaque assay, put it in the incubator, come back the next day and look at it. And so this measurement of virus is an asynchronous kind of thing. You have to then, you take samples, and often what you do is put them in the fridge. And then once you've got all of your samples together, then you do plaque assays on every single one of them. Yeah?
Is there any growth curve experiment where you're not going to artificially lyse the cell? Because isn't that the only way you can tell the difference between the latent and eclipse period? Okay, so Ian's getting ahead of us a little bit here. So latent versus eclipse period. And so we've talked a little bit about eclipse versus latent period here. We've got eclipse at the top and latent at the bottom. The latent period is you haven't done any extra messing around with the cells at all. But if you do that, then you can't necessarily define when you have an eclipse period. And so the difference between the eclipse period and the latent period, so basically latent period minus the eclipse period, in some cases you'll have some time, again, on the x-axis here, where you'll have perfectly infectious virus particles, but none which have actually been released from the cell. And so it's the eclipse period that you get those perfectly infectious virus particles. In some cases, you have to build up the number of virus particles inside the cell, or, as we talked about for MS2, Phi-X174, T7, there are lysis proteins that build up on the inside of the cell. There are infectious virus particles inside the cell before these lysis proteins allow them to be released. And if you really care about how long it takes to get an infectious virus particle, then you have to do the external lysis to the cell. So in most cases, actually, you don't do an external lysis because it's an extra step that you have to add to your experiment. Okay, more questions on eclipse? Yeah, Angela.
So you just find what the absorption period is through experimentation, like when you would dilute or wash?
Okay, let's not get into that right now. fascinating, wonderful, important question, but somewhat irrelevant for most of the people here, other than I think one, two, no, a few of you. Yeah, it's what happens, how you actually have to make sure that you have this complete infection. And so that's this process right here, which for some viruses turns out to be a real pain. We'll just leave it at that. Okay, so hopefully, oh, sorry. Do you have a question? Yeah. Okay, so here we are. See, well planned. So important things, yes, absorption is an issue, how long it takes and how stably the interaction is. But the key, again, for these one-step growth curves is you have as close to 100% infection as possible. And because you've got cells floating around and virions floating around, sometimes you're going to have 2 that associate with 1, sometimes you have 0 that associate with 1, etc. And so the basic idea is if you have a gazillion viruses and very few cells, yes, they're all going to be infected. But usually you have a limited number of infectious virions which are around. And if you need to do this wash step or dilution step to make sure they don't reinfect later on, then you want to make sure that you have enough but not too many of your virus particles. And so that has to do with the Poisson distribution because they're just individual particles that are interacting with each other. And the idea here is that the Poisson distribution, so the proportion of cells that are associating with and assuming infection, assuming you've got absorption and everything working out properly, with a particular number here. So say the proportion that are infected with 1 virus particle is relative to E, just the mathematical constant, the minus m to the k, again the number of particles here, divided by k factorial. And so this is just the description of how many virions you have per cell. And the important thing here is the m. Actually, I probably should have made this a lowercase m. And MOI is just literally the number of infectious particles divided by the number of cells. And if you have an MOI of 1, you can do the calculations here. So, and most importantly, usually people care about the number of uninfected cells. So P of 0 at an MOI of 1 ends up being about 36%, which means if you just mix the same number of viruses and the same number of cells, 36% of them are not going to be infected. So your one-step growth curve is not going to be a synchronous infection by any stretch of the imagination. So that's all that this is really about. Would I ever ask you, say on Monday, for instance? Do the Poisson if I were to give you something like this. And if you look at some of the old exams, you'll see this. I'll give you a table, which would have all of the calculations for individual M's and individual K's. So that's, and I've done that in many years, yeah.
But would you expect us to remember what PK is and all that without any kind of a,
Oh, so yeah, so the proportion, yeah, so what this means,
right.
Yes, I would expect you to know that. And usually, actually, if you go back and look at my old exams, almost always I'll give you this formula.
What if you don't remember how to do
Pardon?
What if you don't remember how to do natural logs and factorials? Quick tutorial?
I would study it over the weekend. Just a suggestion. Okay, so I was about to say that's all the math we're going to do. That's not true because we haven't gotten to our T numbers yet. So all the interesting stuff, again, as far as I'm concerned, is happening here in the latent phase. And this is also a nice review talking about latent versus what's going on in the eclipse phase. So just to skip ahead a little bit, the eclipse phase gets you to here, where you have infectious virions. Getting from here to here, that would get you to the end of the latent phase. So all the interesting stuff, again, virion binding to receptors on the outside of the cell, genome being released, lots of different ways that this can happen. You have expression of the early genes, which are almost always involved in replicating the virus genome, But we also talked about some examples where this also regulates something going on with the host. So turning off of the host restriction endonucleases in the case of T7. Turning down the host RNA polymerase, again, in the case of T7. And we'll see this happening in many other cases with the other viruses as we move on through the rest of the term. Virus genome needs to be replicated. And then your late genes, which are almost always your structural genes, are the ones which are made from new genomes. And these new genomes then lead to production of messenger RNA. Cellular translation takes place. These proteins get together with the nucleic acid, make your virions, the assembly process, and then these guys get out from outside to outside to go out and do and repeat this whole cycle again and again and again. Sorry. Right.
And so, if you're looking at the difference, if you want to see the difference between your eclipsed and latent phase, that's basically the difference between, if there is a discrepancy between the assembly of the virions and exit?
Correct. So the difference between your end of the eclipse period and end of the latent period is when you have these infectious viruses being produced outside the cell. And the only way to tell what the eclipse period is, getting back to your earlier question, is by doing an extracellular lysis. So to know when this has actually happened, unless you have a way of looking inside the cell, which you can do, there are other ways of doing that. But in a classic one-step growth curve, you would be breaking open the cell before these guys have actually been released. Okay, so what is this genome? What's being released? 6, although many people will say 7. I don't agree with the 7. I think that the 7th is actually a subset of the 6th. But Baltimore had 6 classifications in terms of what nucleic acid is packaged inside the virion, what's in this guy right here, and how it undergoes, this replication step, replication of the virus genome. So you have 2 major flavors, if we want to call it that. We've got those that have RNA genomes and those that have DNA genomes. Double-stranded DNA is easy because DNA-dependent RNA polymerases, lots of cellular DNA-dependent RNA polymerases, make the messenger RNA that gets translated into protein. So just like cellular genomes with these double-stranded DNA. You also have, and this is that extra one here, single-stranded DNA, sometimes a mixture of single and double-stranded DNA that's packaged in virions. I can think of this really as a subset of your single-stranded DNA. Single-stranded DNA has to be copied into double-stranded DNA before it can be transcribed and translated. What example did we talk about here so far? Thank you. FIX-174. Man, if the chance of the science march tomorrow are that slow, we're going to have trouble. So FIX-174 down here. Then we also talked about one of these kinds of viruses. Which one would that be? MS2, thank you. So these are viruses that package single-stranded RNA in their genome and single-stranded RNA of the positive sense. Positive sense just means this RNA, once it comes inside the cell, can be translated immediately by the cellular ribosome. These turn out to be incredibly common in plant viruses. Then you also have these other kinds of RNA viruses up here, and then we'll get back down to this one at the very end. So negative strand RNA viruses and double strand RNA viruses. Negative strand RNA viruses have a big problem. And it turns out the same thing is true for the double-stranded RNA viruses as well. And that is that this RNA can't do anything. It comes inside the cell. If you just have that negative strand RNA, which is there, nothing happens. It can't be translated because it's the wrong strand. It's the wrong coding sequence. So any of these negative strand RNA viruses have to have a RNA-dependent RNA polymerase protein that comes in with the genome at the same time as the genome is released. So they're absolutely dependent on a protein coming in as well as the RNA. And this clearly can present some issues if, in terms of getting that nucleic acid inside the cell, if you also have to be bringing a protein along with it. Turns out that double-stranded RNA viruses also do this. In theory, they wouldn't have to, but in practice, they do. So the double-stranded RNA genomes also are bringing in their own RNA-dependent RNA polymerases. So these are all the kinds of viruses that were known before. Baltimore and Temin published their papers also in the late 1970s, where they saw these retroviruses. Retroviruses, in the classic case, encapsulate single-stranded RNA of the positive strand, but instead of doing this and being translated directly, like we saw for MS2, all of the Levee viruses, these now have a reverse transcriptase that makes for single-stranded and then double-stranded DNA, and that then gets translated, or say transcribed, then translated into messenger RNA. Yeah?
You might be getting to this anyway, but the single-stranded and double-stranded RNA, they bring their own protein with their own RNA-dependent RNA polymerase. Like, they replicate immediately when they get inside the cell so that they can produce messenger RNA. The single-stranded RNA can immediately produce messenger RNA, but do they still replicate immediately, or does that happen later for them?
Okay, so the question basically has to do with timing, is when you do genome replication versus when you do messenger RNA production. And we'll talk about that when we talk about the negative strand RNA viruses later on. But in general, it's going to be first you need to make messenger RNA. And it turns out that there are some advantages to being a negative strand RNA genome. Is the RNA-dependent RNA polymerase, when it copies this, is already making positive strand. So that can immediately be translated, as opposed to having to make the positive strand, then the negative strand, then the messenger RNA. So usually what happens is the messenger RNAs are made first, and then there's a switch that goes between making those messenger RNAs and making genomes, not unlike the translation versus replication that you see in some of these positive strand RNA viruses like MS2 and the Levee viruses. So hopefully that's not too long-winded to answer your question. Okay, so we talked about the genome that's on the inside. What about the outside? This has to do with geometry. Again, the other part of math as far as this course is concerned. So hopefully everyone is clear with icosahedral symmetry. We've got 5-fold axes of symmetry. Here they are. 3-fold axes of symmetry. And turn it around a little bit. Actually, that's the easiest way to look at it. 2-fold axes of symmetry. So 180 degrees and 120 degrees, etc. All of those to give you back your same particle in the first place. Most of viruses, in terms of packaging their genome, just having one capsid protein and 60 copies of it is just not big enough in order to encapsulate all of that nucleic acid. So in order to have a icosahedrally symmetric particle, you use the quasi-equivalence, which is where you have your coat proteins, here conveniently all of these little commas, which then can either come together as 6 or as 5. And how these 6s and 5s fit together, there are a limited number of geometric ways that this can happen. I've posted a couple of links to that on D2L if people are interested in getting into it. But the best way of thinking about this is the triangulation number. And so it's getting from one 5-fold axis of symmetry, these green pentagons, to the next 5-fold axis of symmetry via these 3-fold or 6-fold, because they're 6 copies right here. And so here, a t equals 1 is just one step. T equals 3 is one step in one direction, change and go in another direction. T equals 4, 2 steps in the same direction, but the easiest way to look is in the middle, here, 1 step, 2 steps, etc. If I were to give you something like this in the exam, I wouldn't give you a nasty cryo-EM reconstruction of these viruses. I'd give you a nice pattern, something like this, which is a nice T equals 12 quasi-equivalent icosahedron. If you're interested in coming and looking at it, I'll have it up here after class to take a look at. So what I would do is not give you something like this. I'd give you something like this in order to try and calculate what that is. So people happy with this? Less happy with it? Will be happy by Monday?
I'm super happy.
Super happy. Good. Super happy. I really like that. So, of course, there are exceptions. It's biology. There are always exceptions. This particular one way that you can tell that it's an exception, if you just look at the number of capsid protein subunits. How do you know what the T number is if you know the number of capsid proteins that are in there? Divide by 60, exactly. So if you divide 360 by 60, what do you end up with? 6. I challenge you to use whole numbers and have h squared plus hk plus k squared give you 6. So it can't work. And it turns out that the reason for this is instead of quasi-equivalence, I like to think of this as quasi-non-equivalence, because each of these different single molecules of exactly the same sequence has a slightly different structure, as opposed to just 2 different structures, which is we have with quasi-equivalence. Now you've got 6 different structures. Those are at the 5-fold axis, and each of these guys at 3-fold axes. So same protein, 6 different structures. Wait a minute, that's not what Bartlett said in Principles of Biology, and if you've had that before. So, but that's still the basic idea. That being said, these still overall have a very similar structure, which is this beta barrel structure. So lots of beta strands lined up with each other in this wrapping. And we'll talk a little bit more about that a little bit later on. I brought one of my structural models today, which actually has 2 of these beta barrels right next to each other. So beta barrels extremely common in terms of the structural subunit on how these come together. Yeah?
Would it be fair to say that their structure is functionally the same? I mean, if they can form those 6 different structures, but all of the structures, like the one protein can form the 6 different structures, but functionally they're doing pretty much the same thing?
Yeah, so okay, the question is basically sort of structure versus function thing here. But yeah, the capsid protein, what is the capsid protein doing? It's making a capsid. It's making it through having 6 slightly different structures relative to each other. But it's still a capsid protein, and that is its function, is they're making the capsid. Okay, so I think I didn't do a good job talking about these the first time around, so I wanted to revisit this really briefly. Talked about the virions right at the beginning. We're talking about enveloped virions here. So the envelope is, again, just this lipid here. We've got the inside of the virion here, the outside of the virion here. This can come from basically any kind of internal membrane that you can think of on your cell, or the plasma membrane. But in those, all of these enveloped viruses have some kind of virus protein that's sticking through this membrane so that the virion can associate with some poor other host cell that it's going to get into. Usually these are glycosylated. That's what these little Y structures are supposed to mean here. Very often they also have disulfide bonds. That's what these green bits are here. Often multimeric structures. We saw some of the trimeric structures already before. This is one of these dimeric structures. And the vast majority of the protein is usually present on the outside, with a little bit which is actually present on the inside here, tied into the structure. Yeah, Alice?
Does infection cause different glycosylation patterns in the host cell, or is it usually that they just pick up the host cell membrane as it is and they don't usually encode differences?
Okay, I'm not sure I completely understand your question, but I'll do my best to answer it. So basically the question is, Where does this glycosylation come from? Where does the glycosylation that ends up on these virus glycoproteins? Is it a cellular glycosylation process or is it a viral process? And the answer is yes. Some viruses have their own glycosylases, and some viruses are just using the glycosylation process of the host.
Is there a hybrid, though, where they transpose?
Oh, so the question is, are there some where you have both cellular and viral? Yes, definitely. So yes and yes and yes, maybe, is a better way to answer that. So how you get these membranes wrapped around your nucleocapsid or the nucleic acid associated with proteins we were talking about before. In some cases, again, you do have, like we looked at right at the beginning, very first of these slides, you have a pretty regular icosahedral capsid that's got the genome on the inside. That then will bud out through the membrane and give you all these membrane glycoproteins, but these are all viral ones. And the immediate question that comes up is, well, okay, how does the virion know, or the nucleic capsid know, that this is where it needs to associate with those virus membrane proteins as opposed to the non-viral membrane proteins which are there? And it turns out that a lot of cases here, we've got lipid rafts, we've got the membrane is not by any stretch of the imagination a homogeneous set of proteins, et cetera. And there are usually direct interactions between these virus proteins that are sticking in the membrane, that little bit which is sticking on the inside, that then is going to interact with either this capsid or in some cases just the nucleic acid and the proteins that are associated with each other. Either it can be direct, there can also be a matrix protein, which is between these two kinds of things, kind of serving as an adapter between these two. Some of these proteins also have fatty acids which get attached to them. Why fatty acids attached to them? Because then they're going to be targeted to the membrane. And in fact, a lot of the membrane targeting that we now know from cell biology, which a lot of it was originally discovered studying virus proteins, but these fatty acid tags will lead to specific parts of the membrane, and that's where you're going to have this virus assembly and coat protein process. I want to talk really briefly about virus taxonomy, because one of the viruses that I discovered. So a little bit of tooting my own horn here. But the idea, and this is why I brought this structure with me, is that if you want to think about virus phylogeny and virus taxonomy, what most people are doing now is they're starting to think about the structures, which, anybody go to the biology seminar yesterday? I was there, talking about how to classify biodiversity, talking about measurements of skull sizes, et cetera. So this is sort of the equivalent now for viruses. People always used to think about using molecular tools, just the sequence of comparisons. Now people are thinking about using the actual structures of proteins in order to think about really deeply branching viruses. And the idea here is that even if you have viruses that infect 3 completely different domains of life, this one is infecting archaea, this one is a virus particle that's infecting bacteria, this one, adenovirus, that we'll talk about after the midterm, is infecting humans. They all have basically this identical capsid protein structure, even though they have no obvious sequence similarities to each other whatsoever. And so we think that's evidence for some kind of common ancestor, and it's not just me. There are other people who think this as well. Again, we have a virus that's infecting archaea, bacteria, another bacterial one. This is one that's infecting a eukaryote. These are other eukaryotes. This is actually one that infects other viruses, Sputnik, that we'll talk about later on. Here are those adenoviruses, all of which have this same basic structure to them, which again we think is evidence that these are all related to each other. All of these virions have picked up a capsid protein and then modified it over the very, very large number of generations before we get to here. Yeah?
Are they similar in morphology or morphology and nucleotide sequence?
So they're just similar in terms of the morphology of the protein. There's 0 nucleotide sequence similarity and practically no detectable amino acid sequence similarity. But the proteins are basically superimposable on each other. which is really kind of bizarre. And again, it could be convergence, could be that you're focusing in on something that only makes this structure, but we saw SV40 clearly is doing something else. So there's some other ways of solving the same problem. But this, I think, is the most parsimonious argument. We'll never know until we invent time machines.
Does this tell us that the host proteins that they're recognizing are going to be very similar as well?
So the question here is the host proteins that they're recognizing also going to be very similar? It turns out that in all of these cases that I've talked about, the actual receptor interaction is very different. It's actually a separate protein which is doing that. And it's true for many, many, many of these. So the receptors are going to be different for these different adenoviruses, different for STIV, different for PRD1, different from PM2. So the overall structure of your virion is going to be extremely well conserved in terms of just the structure. The actual interactions with your receptor are going to be very different. And that makes sense from an evolutionary point of view because the cells probably don't want to be infected. The viruses want to infect the cells, and so it's going to be this evolutionary pressures. And people have done some great experiments looking at that, and you do see much higher rates of evolution in the receptor binding proteins and the receptors, if they're proteins, how they're interacting with each other. A little bit of an aside here, but we'll talk more about that when we move along. The other aspect of taxonomy, which is really changing, literally this was this year, is now incorporating a lot of those sequences from the little green dots that I talked about in the very, very first lecture. So massive numbers of sequences which are there. How do you incorporate them into some kind of taxonomy? What do you call a virus species? Which is something I talked about with the seminar speaker from yesterday in biology. What do you call a seminar speaker? He works with bats. So bats versus looking at virus sequences. Defining a species here is really, really hard. But if you have enough sequence differences, now the ICTV is thinking about using those now, and particularly these kinds of phylogeny. The other thing I wanted to point out here is that the sequence diversity which is out there, all of these little twigs, way overwhelms what we know about, which are these kinds of viruses here. And interestingly enough, these viruses that we know something about, again, these wedges here, turn out to be extremely similar to FIX174. So these single-stranded DNA viruses that replicate through these rolling circle mechanisms. Okay, sorry we're skipping around a little here. We did, trying to get through a whole 3 weeks in an hour and 5 minutes. So here is classic example of a receptor binding protein, also a membrane glycoprotein. What you don't see down here is the transmembrane domains, which are actually down here at the very bottom here. These structures were all solved on the just extracellular part of the influenza protein that does actually do hemagglutination, why it's called HA, hemagglutination. It's produced as a precursor protein, HA0, so the red and the blue are covalently attached to each other. There's one long polypeptide. Then you have proteolysis that takes place. Proteolysis is a really common occurrence in terms of virus assembly. And one of the main things that it really seems to do is give you that metastable state. So you remember virions have to be very stable on the outside of the cells, yet they still need to be able to release their genome once they get to an appropriate host. So this kind of proteolysis is an extremely common occurrence. What happens with the proteolysis in this particular case, here didn't have any N and C termini before. Now we've got two separate proteins, which are now labeled as blue and red. These then undergo major conformational changes at low pH. Once that virion gets into the endosome lowers the pH, you have a conformational change, and it's those conformational changes that really lead to membrane fusion. And we saw a couple of examples. We talked about HIV, we looked at the dengue, how all of these membrane fusions take place due to conformational changes and the presence of these so-called fusion peptides a little bit hard to see here, but there's a pretty clear change if you look between this structure, a couple of beta strands, and the alpha helices over here, particularly this one extending there. That's what gets inserted into the host membrane. And that insertion to the host membrane is the first step that is going to lead to the fusion of the two membranes and the release of the genome, which is on the inside down here at the bottom. And we'll look at this again when we talk about some more of these envelope viruses a little bit later on. Okay, questions on sort of the general stuff. Now we're going to talk about the Levee viruses, the microviruses, and the podoviruses. Levee viruses, single-stranded, positive strand RNA viruses, which are infecting bacteria, very simple genomes, maturation protein, which really has much more to do with interacting with the receptor, which is what in E. coli? It's the F. pilus pulling that virion inside the cell. The coat protein gene just makes lots and lots of copies of it. Yeah?
So should we assume that that's, at least for the purpose of the test, if questions are asked like that, that it is the F. pilus pulling it inside the cell? Because the book is kind of dubious about how the RNA actually gets inside the cell.
Yeah, so, okay, question is, believe Stedman or believe the book? No, sorry. So if I were to ask a question like that, which I think I have written some questions, I haven't decided which ones to use yet, then I would try and make it clear that that's the proposed mechanism for getting inside the cell, rather than absolutely that's what I said, and so that's what it is. So yeah, I would try and be as clear as possible about those things. So yes, maturation protein certainly is involved in the virion associating with the pilus. That is clear. Whether that's the actual entry mechanism, probably. We can't think of any other real way to have that happen. Although one of the cool things about pili, sorry about this quick aside here, is that they're actually hollow. So maybe somehow the RNA gets into the pilus and goes in that way. Who knows? Yeah.
Aren't there viruses that hijack like in our cells, we have the machinery, like lysis and actin, and I know, like, through our nerve cells, they take it and go the other way.
Yeah, so there are definitely intracellular transport. We talked about it a little bit in terms of entry. This would be extracellular, of course, in terms of actually getting into the cell in the first place. But once the virion's on the inside, this is a nice review anyway, then very often that virion, or at least the nucleic acid in the virion, has to get to the nucleus. And very often that's moving through these intracellular transport mechanisms, microtubules, actin, etc. And there's a really cool video that the BBC did with these things walking along these things. So, but we'll talk about that more later. So coat protein, you need lots and lots of coat protein. And then, of course, it's an RNA virus, so it needs an RNA-dependent RNA polymerase. And then, being that this is a bacterial cell, got a pretty tough membrane on the outside. In fact, two of them in this case. It's infecting E. coli. Has to be some way of getting out. It's the lysis protein. This is encoded in MS2 and a different reading frame. And so it's an overlapping reading frame. You're using this piece of the genome multiple times in terms of different proteins. And yes, answer Ian's question already, the lysis protein can have lots of mutations in it and still serve in the same way, because it's basically just serving as a hydrophobic thing to make a hole in the membrane. So yes, you can have lots of mutations here, not causing problems. You can't have very many mutations here because it's an enzyme, and so this is going to be much better conserved than what's down here, which kind of makes sense if you look at some of these other single-strand RNA viruses, very different in terms of how they get out here. How do you make these different proteins, and most importantly in different amounts? It's all about the translation, because it's just the RNA, positive strand RNA. That's all you've got. How is that regulated? It's regulated based on secondary structures. Secondary structures which will either allow the ribosome to associate and translate that piece of RNA, or secondary structures which will block the ribosome from making these particular secondary structures. And the best example, the best understood of these, really has to do with the amount of the coat protein, which starts over here, and the replicase protein replicose protein can't start to be translated until you have translation of the coat protein. So coat protein gets translated first. You make lots of coat protein. Then you start to make the replicose protein. And eventually the coat protein gets shut down because the coat protein itself starts to block more translation of that actual coat protein. So lysis protein is a slightly different story, and that has to do with the ribosome moving back and forth on the RNA once it gets to a termination point. We'll talk about ribosomes staying on RNA for other viruses a little bit later on. Frame shifting, for instance, bounces back and forth. Read through, have a termination codon that actually is not used as termination. The ribosome continues. Many viruses take advantage of the fact that this ribosome doesn't come off immediately right after you have translational termination. And in the case of MS2 and the other viruses that use this particular kind of regulation, but mostly MS2 here, it's just the ribosome bouncing back and forth along the RNA and occasionally, about 5% of the time, getting to this particular point. How do people know this? They just put more or less extra nucleotides between these two. You had more nucleotides between the start of the lysis and the stop of the coat. You had less production of lysis protein. You had a shorter distance between the two. You had more production of lysis protein. The maturation protein, again, mostly this pilus binding protein, is made only from new genomes. And the reason it's made from only new genomes is because the secondary structures that form on newly made genomes are different than the secondary structures which are formed on older genomes. So it's alternative secondary structures. This one allows, translation of these proteins, and this one does not. You have a question? Is stretching.
Yeah, I've got
Sure, yeah.
a question about the lysis protein.
Yeah.
Do you think the location of the start codon is relatively conserved, so that, like if it's too close, like lysis would happen sooner?
Yeah, so the question basically has to do with, again, evolutionary questions. And so presumably it's now optimal for getting lysis to happen at the right time after you've had production of enough genomes, enough coat protein, enough maturation protein in order now to have enough infectious particles. And one of the things that I didn't emphasize when we looked at it, when we looked at some of those micrographs where we had infection by some of these small RNA phage, they were just hovering everywhere. So you get very, very large burst sizes. The number of virions per infected cell is very, very large with some of these, which is partly why you have so many of them in the sewage, is because these numbers are ridiculously large. But if that lysis protein start was actually a bit closer, you'd actually have fewer of them. And you have fewer of them. If it was further away, you might even have more, but then at that point, maybe the cell isn't going to be able to replicate enough of these things. So it does seem to be an optimal situation. And if you look at these viruses and some other related ones, they are pretty similar to each other. So F2, for instance, which we didn't really talk about, the first one discovered, very similar kind of lysis recognition here. OK, so more questions, on these small RNA phage. So switch gears, talk about DNA viruses. These are the microviruses. The main one we talked about is Phi-X174, but there are many, many, many more of these. Again, those CREST viruses that we looked at in the metagenomes right at the beginning. Many of these are quite closely related to all of these other viruses, some of which are parasites of parasites, so the intracellular bacteria that also have these viruses associated with them. The genome here, the big surprise, the message from outer space, is that we have lots of overlapping open reading frames, but to be able to get away with having these overlapping open reading frames, they have to be very resistant to mutational change. So B, you can mutate like mad. E, K, and A are not absolutely required for virus function. So yes, you can pack in a lot more extra genetic material and make more proteins, but they've got to be pretty flexible here. And it turns out that actually most of the microviruses don't have all of these extra overlapping reading frames. 5X174 is a bit of the exception as far as that's concerned. The other thing here I just wanted to also mention is that, not surprisingly, all of these structural proteins, J, F, G, and H, these do not have overlapping open reading frame because you can't make massive numbers of changes in there and still get away with it. Yeah?
Mine is specific to A and A asterisk.
Right, A star. Mm-hmm.
If a mutation happens in the part of A that is not star, is that bad? I mean, is the first half of A structure? Why make the difference? Why make the separation and not call it A and B?
Okay, why? Evolution. So yeah, no, the answer is we really don't know. We know that an A star protein is made, so it's definitely there, but you don't seem to need it. And if you go and look at these 5X174s, it's definitely there. And the question really is sort of why, and the answer is we really don't have a good handle on why exactly it is.
But A proteins always include an A star.
They do not always include an A star. So 5X174 does, but many of the other microviruses actually do not have an overlapping open reading frame there. And even this B protein, the internal scaffolding protein, is also not commonly present in a lot of these other microviruses, which are there. So this is the excuse me, big surprise since the publication of the chapter in our textbook. It's the H protein, which, if you just look at virions, there's no obvious place that this H protein is. They're nice symmetrical virions. And then as soon as they associate with the LPS on the outside of the cell, this H protein makes this sort of one-way ticket whereby the genome is transported through that into the host cell. As I mentioned when we talked about the different kinds of nucleic acid that are packaged, single-stranded DNA viruses have to become double-stranded in order to be able to do anything. And when I say do anything, that means transcription, leading then to translation. That all happens with cellular proteins. The A protein, which you do absolutely need, also known as these the cress part of the cress viruses that we looked at before. All of these proteins, they will cut one strand of your double-stranded DNA, and with helicase activity, move that non-template strand out of the way and allow the polymerase to just go around and around and around. Each time it gets around once, this A protein reverses the cutting activity. What's the reversal of cutting activity? Ligation activity. So it ligates that piece back together. But the polymerase keeps going around and around and around. These single-stranded pieces can either be packaged into virions or they can become double-stranded pieces later on. So T7, any real quick questions about T7? I'm not going to go through the slides because we're done. Good luck on Monday. Any more questions, please email me over the weekend or come find me at the March for Science tomorrow morning.